Yeah, to continue from the previous video, there are a few other things we need to know first before we continue with the docking. And in this case, we're going to first try to know the grid center to which the co-crystallized enzyme, uh, ligand, sorry to say, was docked in the receptor. One easy way to do this is to use Discovery Studio Visualizer. And from here, we can open, just browse to your working directory. So once we are here, we can still pick this, and then we're going to see the whole thing again. Interestingly, you can also use this as a means to clean the protein or even extract your. So quickly, we'll learn how to use this tool. Right click. So if you right click, change mouse mode to rotate to make it easy for you to flip through the enzyme. We can come back here again to change the background color to white for better visibility. Okay, and then click this arrow, and like we saw just now using the other tool, Autodock, you can see everything here too, and you can delete them one after the other. Um, Ligand groups. We're interested in ligand groups because we need to know the grid parameters. So this is chain A, chain B. We want chain B out. We want chain C out. So using the same process, we have deleted all the other components, the monomers and their co-crystallized ligands. Now, how do we get the grid center? It's quite easy. We have the options here. Define and edit binding site, and we have three options in DSV. We can use receptor cavities, PDB site records, and then from current selection. Yes, they can all be accurate, but since I have a co-crystallized ligand, I normally prefer to manually select, which is what we saw here now. Select from current selection. Trying to get a clear picture. So we will just click this edge, for example. It turns yellow. Then hold your shift button, click this other edge, then just pick all the edges out so that you can capture around. Yeah, feel free to flip the whole structure around so you can capture some more edges. Hold your shift again and continue. You can see that all the edges I am clicking, they are turning yellow. That is, they are being highlighted. So once we have done that, you will notice here that from current selection is now also highlighted. So we can just now click this and you see it has built a grid around it, SBD site sphere. So if you click this, when it's now this dark ball, you can simply get the attributes from here. So you have the attributes listed here. This is your XYZ. So these are the coordinates. You take note of these values. X 88 point this, Y and Z. You can save the attributes to somewhere you can get it later. So this way we now have this attribute. But because we are using a ligand that is co-crystallized, we're trying to ensure that it is the same ligand that was previously docked that we are extracting and docking again, like I mentioned in the first video, because we have validation in mind in this case. But for your routine studies, it could be docking of compounds from PubChem or any other site. So you could use the co-crystallized ligand to identify the active site, and then from there onwards, use those parameters to set the grid for the receptor ligand interaction. But in this case, I want to extract the ligand and I want it in Mo2 format. If I use DSV, it will give me a PDB file, but a Mo2 format is preferred. Basically, you could use one tool to do all of this, but we're going through all different tools to see how to either use either of them or use them together in combination. So Chimera X is also another interesting tool, and what we have here is simply the, I can just use this command line to change directory to my working directory already, which I have done here. So you can see current working directory is now training videos. So if I come here and I say open, I think I'm in the wrong directory. Yes, once we open, it's supposed to take us to the actual directory that was set. Now I'll pick this again. Like I said, I just have a copy in case I need to come back to get information. So open, and this is the complete structure all over again. And we want to delete B, C, D, and all the waters. So we can use the command line to easily select, for example, select, bad spelling, select space /B, then enter. That selects B automatically. But if you want to even go further, select space /C. So you see we can select multiple sections. Let's just now get all the other ones we want to get out done. Select space D. So we have only A left here. You could also do this by coming here, select chains. In this case, it has 4 different chains. So this is B, this is C, this is D, and that's A. Or if you want to select all, you can do that here also. But all we have done here so far is to select them. We could either use the command prompt to also delete, but since they have been selected, we will just now come to actions, bonds, atom and bonds, down to delete. So we have only to move, just right click, hold it down to move this one around, then left click to rotate. Okay, so we were trying to extract the co-crystallized ligand in this case. We'll come back to select residues. So you see we have water, we have EAA, that's itacrinic acid, and we have the standard amino acids. So what we're looking for is this. So to delete the amino acids, we click this. They have all been selected now. Back to atoms, this, then delete. They're out. Then we have water left. We can now come back here again to select residues. We select water, and then action, then this, delete. So we now have the co-crystallized ligand. Okay, now just scroll to push it around to left for the docking proper. Then what we now want to do is save it in our working directory as save. Like I said, there are several formats, but Mo2 is a better one for this. So let's just call it itacrinic acid. Remember not to allow spaces, just because of pi mode we use later. Itacrinic acid in Mo2 format, save. So if we come to our working directory here, we should have, yes, in Mo2 format. So this is how to extract the ligand from the protein. In this case, like I said, because we're interested in validation, but the compounds used for your study can come from public databases like PubChem, Zinc, and what have you. But what's important is we now know how to identify the grid. There are several ways of doing this, but this is a very easy approach